We want to begin our study of the membrane of the cell, talk about the structure of the membrane and its function. We're going to look at the current model, which is referred to as the fluid mosaic model, and we'll talk in detail about the parts of the plasma membrane, the proteins found within it, the permeability of the membrane, and then some modifications we may have to the membrane. Our current view of a plasma membrane of a cell is referred to as the fluid mosaic model. And think about what the term mosaic is. We're talking about mosaic tile. It's made up of lots of little pieces, variety of things. And you can see an image here of the phospholipid bilayer. It's made up of a lot of different things. It has a mosaic appearance. We refer to it as a phospholipid bilayer because it has phosphates and lipids. We have both phosphates here and here and lipids here. And you can see that it is a bilayer. It has two parts, part 1 and part 2. We also have proteins found within the phospholipid bilayer. And these proteins, if you'll notice, are kind of floating around. If you're looking at it from the surface, what you see on the surface is just a small view of the overall protein. Notice the protein comes very deep below the surface and may even be found on the inner surface. Some of these proteins also have a carbohydrate chain attached here. And we also have the addition of cholesterol there. So you can see it's like looking at mosaic tile. We have a variety of things that make up our phospholipid layer. If we look at the phospholipid bilayer in a little more detail, the plasma membrane is common to every cell. If you're a cell of any organism that's living, you have a phospholipid bilayer. It has two jobs. It's going to separate the internal living cytoplasm from the external environment of the cell. In order to separate the internal and the external, we have to line it with heads that are a phosphate but that are hydrophilic. They like or love water. On the cytoplasmic side, we have the same hydrophilic heads. But in the middle, we're going to put between them nonpolar hydrophobic. They fear water. These fatty acid tails, this creates this isolating layer that will separate the living from the non-living sides, the internal environment to the external environment, and you'll see it keeps the water inside the cell in and any water that's outside the cell out, making a selectively permeable membrane. If you look back at the cholesterol found within the membrane, it helps modify the membrane, it helps stiffen the membrane. Without cholesterol, cells would be flabby, they'd have no structure to them. At lower temperatures, it also keeps the membrane from freezing because it doesn't allow the phospholipid tails to actually come in contact with each other. It keeps them pulled apart, keeps the membrane fluid. But cholesterol is necessary. We do need it to give some rigidity, some structure, help stiffen that membrane. We also see that the membrane has proteins. Now, both lipids, the fats, and the proteins can have a carbohydrate chain. The carbohydrate chains act as the ID tag. They are what will ID your cell from my cell from your cat's cell. Now, the carbohydrate chains come in two forms. They can be a glycolipid, that's a carb chain attached to the fat, or they could be a glycoprotein, that would be a carbohydrate chain attached to the protein itself. Because if we looked back at that phospholipid layer, we have our phospholipid, but we also have proteins embedded in. A carbohydrate chain attached to the phospholipid portion is called a glycolipid. A carbohydrate chain attached to the protein portion is called a glycoprotein. And again, they act like your ID tag. They're your fingerprint. That's why we cannot take a liver out of a stranger and give to you without making sure that it has similar glycoprotein and glycolipid chains. Your body uses those chains to ID your cells and therefore reject or attack anything that's not you. These proteins here may be either peripheral on the edge. You can see them both on the outer and the inner edge, or integral. Now, peripheral proteins are going to be found only on your inner edge, whereas integral are integrated. They're embedded completely throughout the membrane. You may see them sticking out both the top, all the way through the layer and through the bottom, whereas peripheral we're only going to see on that inner edge. Look at these proteins a little more detailed because we have more than one type. We have a channel protein. It's like a tube that can allow the passage of materials through it that are notably very small. It's not a huge tube, but it will allow particular ions, sodium, potassium, to freely flow in and out of an otherwise selective barrier. A carrier protein. Now, a carrier protein doesn't stay open like a channel protein. Instead, a carrier protein opens up, accepts a molecule, and then dumps it out the other side. It actually becomes a part of that molecule. It interacts with that molecule and it takes it in and then it dumps it out the other side. A cell recognition protein is the protein I was talking about earlier with the carbohydrate chain. Here's a protein embedded throughout the membrane. It has a carbohydrate chain attached. This is your ID tag. It helps your body recognize what is you and what is foreign, what is not you. It also causes your cells to respond. Now, we have some particular types of cell recognition proteins. One we have in the human body is called the major histocompatibility complex, whereas histo means tissue. This is the protein responsible for identifying tissues so that your body knows if that liver we've just surgically put in you is your liver or a liver from another organism, or another living thing. If that ID tag doesn't match, then your white blood cells will go in and attack that. That's why someone who has an organ transplant must always take anti-rejection drugs. Without those drugs to depress the immune system will very quickly figure out that that organ is not of the original person, and they would attack that. We have receptor proteins. Now, receptor proteins have a unique shape, and they are shaped so that they fit only a specific molecule. And this is like a lock and a key, that this cell will only respond to a message when this key is inserted into this lock. And it allows the cell to respond to only that specific message. We also have an enzymatic protein. An enzymatic protein allows a metabolic reaction to be sped up. It catalyzes it or speeds it up. It allows a reaction to happen faster than if the enzyme were not used. And we also have a junction protein. So here is one cell, and here is another cell. They each have their own selective plasma membrane, and we want to join them together. So we would use a junction protein that not only holds the two membranes together, to hold the two cells together, but may also allow materials to pass from cell A down here to cell B. So let's look at its permeability. We say that a plasma membrane is differentially permeable or selectively permeable. What that means is it's picky. It will only allow certain things to move across the membrane, and other things are going to be rejected. The things allowed across the membrane are selected by size, by charge, and this means electrical charge. If you'll think back to your ions, like sodium being a positive ion or chlorine being a negative ion, that alone can restrict its movement. If it is lipid soluble, if the temperature is not correct, the pressure, the currents, all of these are factors in what may or may not be allowed to move across the plasma membrane. The other factor that decides if something can move across the membrane is, do we have a concentration gradient? Meaning, is there an area where there is a high concentration and then an opposing area where there is a low? Because particles are only going to move from high to low. We say there is a concentration gradient. So if we're going to move things across the membrane, we have a couple of ways to move them across the membrane. We have two main types. We have passive transport, and then we have active. So we're going to look at passive first. If you think about the term passive, someone that is passive, they're usually very mild, very meek. You don't see them exerting a lot of energy. Passive transport follows that same principle. We don't have to expend any energy for the materials to move. There's no ATP needed. Materials just move because there is a concentration gradient. We have an area of high concentration, we have an area of low concentration, and the materials move from high to low. Now, most often we're dealing with a solution where we have two parts to it. We have our solute, which is our solid, we looked at in the chemistry chapter, and then we have our solvent, that is our liquid. As long as they're concentration gradient, materials will move through. So we're going to look at two types. Diffusion, and we're going to talk about a special type of diffusion called osmosis. And then we're going to look at facilitated transport. Start with diffusion. Diffusion is simply the movement of molecules because of that gradient. Movement from an area of high concentration to an area of low concentration. Now, materials will not continually move. They can only move until there's an equilibrium, meaning when the high and the low are balanced, diffusion will stop. Lipid-soluble molecules, gases, all move by basic diffusion. If you were to stand in the front of the room and spray Lysol, right when that Lysol comes out of that can, that is a high concentration. It will slowly diffuse through the room, and within a few minutes you'll smell that Lysol all the way in the back of the room. That's basic diffusion. The example you're seeing here, putting a crystal of dye in the water. If you will allow that, it will diffuse until the particles are equally distributed. You can take a cup of water and put a single drop of food coloring in it. In the beginning, the food coloring will be concentrated in a little ball, but you let that sit a few minutes and it will diffuse throughout the cup. It'll move from high concentration to low concentration until we've established an equilibrium. Now, water also diffuses, but we call it osmosis. Osmosis is diffusion, but it's diffusion of water. And it's diffusion of water across one of these membranes that is selectively permeable. It's also going to move because there is a concentration gradient. There must be an area of high concentration of water and an area of low concentration of water. Now, what makes this unique is that water can diffuse both ways, meaning water can diffuse into a cell and water can diffuse out of a cell. So regular diffusion is typically in one direction from high to low, but water can be highly concentrated outside of a cell and move into the cell, osmosis. And then the water inside the cell, if highly concentrated, can turn around and have osmosis and diffuse out of the cell. Water is going to go in both directions. It creates a pressure referred to as osmotic pressure. And water moves in the area where osmotic pressure is the greatest. If osmotic pressure is the greatest, water is going to diffuse into that direction. When water moves around, we see a couple of conditions. One, we see that the strength of the solution changes. Because if you have a solution, which is made of a solute, a solid, and a solvent, a liquid, and remember that water is our universal solvent, then there's a strength to that solution. There's either a lot of water to a little solute, or a lot of solute to a little water. Think about making Kool-Aid, where you put 4 cups of sugar into 2 cups of water. That's going to be a high-strength solution, very sugary, versus putting a 1/4 a cup of sugar into 4 cups of water. That would have a low strength. We're referring to its tonicity, the strength of the solution. One type of solution we have is called an isotonic. Now, tonicity is the strength. Iso means the strengths are the same, meaning the solute and the water concentration on the inside and the outside are both equal. So water will move in and water will move out, but there's not going to be any net gain, meaning we're not going to move out more water than we move in. Water is freely moving in and out because the plasma membrane does not selectively restrict the movement of water. You can see in an animal cell, you can see in a plant cell, In an isotonic solution, water's moving in, water's moving out, but there's no net gain of water. We also have a hypotonic. All right, tonicity is strength of the solution. And hypo is referring to having a low strength. Low strength of solute. When you're referring to the solution's strength, you're referring to the amount of solute. So in a hypotonic solution, there is a low amount of solute here. What you're going to see in a hypotonic solution then is that when a cell is placed into this type of solution where there is a low amount of solute and there is a high amount of water, when I drop a cell down into this solution, now because there's a high volume of water, water is going to move into my cell. When water moves into my cell is going to get bigger and swell up. Why? Because the solution that I put it in had low solute, hypotonic, and high water. So when my cell gets dropped in here, my cell is going to gain this water. It's going to cause a cell to swell up. Now for an animal cell, that can be dangerous. They can burst. We call it lysis. They can burst because we just have a plasma membrane, and the plasma membrane can get stretched beyond normal limits until the point that it ruptures. But for a plant cell, we see a little different, because a plant cell has the addition of that cell wall, and that cell wall gives it structure and rigidity, but it will also prevent this cell from rupturing. Instead, in a plant cell, we gain something called turgor pressure. And turgor pressure is what allows that cell to swell up and stand upright. In an animal cell, we run the risk of cytolysis, literally splitting the cell, rupturing the cell. But in a plant, we gain turgor pressure. So if you want to remember hypotonic, you might want to remember hypo reminds you of the term hippo. I think about hippos, very, very, very big. Hypo will always cause a swelling, always cause a cell placed in that solution to get bigger. Now the other type of solution is a hypertonic. Hypertonic means high. So we have a solution where we have high solute, therefore we have a little bit of water. So what if we take a cell that has water in it and we place it down into this solution? Well, the cell suddenly now has the higher concentration, and the solution we've put it in has the lower. So water is going to move out of my cell and into my solution, which is going to cause my cell to shrivel up or shrink. We want to kind of remember this by thinking about just hyper. If you think about a real hyper child, go, 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 run, 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 they're very rarely overweight. They're always on the go. They're typically smaller. Hyper is going to cause my cell to shrivel or shrink. In an animal cell, we refer to that as crenation, shriveling up. If we're talking about a plant cell, it's called plasmolysis, because the plant cell is literally going to shrink the cytoplasm and pull away from the cell wall. You'll see this big gap between where the cytoplasm used to sit up next to the cell wall and where it doesn't. This cell itself is not actually going to shrivel, because the cell wall will maintain its shape for it. Now, this is what happens when you salt a slug. If you've ever gone outside and poured salt on a slug, think about what you've done. You've put a solution on that slug that's very high in salt with absolutely little or no water. Therefore, the slug, whose body is high in water, is going to literally dehydrate. The water will move out of the slug into the salt, and you will dehydrate the slug, which kills it. Same thing that happens if you salt a plant. It will cause the water to draw out of the plant and into the salt solution, which will effectively kill the plant. So we have isotonic, everything's equal, there's no movement of water. We have a hypotonic, hypo like a hippo, the cell's going to swell up. Then we have a hypertonic solution where the cell will actually shrivel and lose water. The other type of passive transport, where we're not going to require any energy, is called facilitated transport. Now, we're still moving from high to low, therefore we don't need any ATP, but it does need facilitation, or it needs some help. So in order to transport materials this way, we're going to have to either use one of those channels or one of those carrier proteins. We're going to either need a carrier protein or a channel to assist us in moving this across. Now, what this allows is for a solute to move rapidly across the membrane. Water, some of your sugars, the amino acids will all use facilitated transport to speed the way that it crosses the membrane, even if the material is not lipid soluble. For example, water is not lipid soluble. Think lipids, fat. Water and fats, oils, they don't mix. They're not lipid soluble, but this will allow the rapid movement of that across the membrane. Because we're going high to low, we still don't require any ATP. It is a passive process. Now, active transport is different. Active transport is going against the concentration gradient. It's going from low to high. It's swimming upstream. That is going to require energy. It's also going to require a carrier protein. Now, some sugars that don't passively move, some of your amino acids, your ions, sodium, potassium, and such, they're going to use active transport to move materials across that selectively permeable membrane. Some forms of active transport. We have regular active transport, move a carrier protein across the membrane requiring energy, and then we have something called bulk transport. Bulk refers to something of larger size, a macromolecule, a big molecule. Well, we need to move large molecules in and out of the cells, and to do that we're going to use a vesicle, this little transportation. They're going to move in and out through a process called exocytosis. Exo to go out, cyte the cell. In this case, this vesicle, which notice also has a plasma membrane, this vesicle is going to merge with the plasma membrane, keeping that selective barrier. We don't want to just open up the cell, because if we did, everything in the cell would go running out. So we're going to merge the vesicle, and when the vesicle merges, we keep our protective layer, and we will allow our material to be dumped out. This is using a phospholipid vesicle to merge with our phospholipid bilayer and dump out a particle that is large. Because it is large, it could not use one of those proteins we looked at earlier. It couldn't use a channel protein. It could not use a carrier protein. These materials are macromolecules. They're too large for that. We may also need to bring materials into the cell. To bring materials into the cell is still going to require ATP. We're going to use the reverse of exocytosis. The particle, when it gets to the cell, will be engulfed and a vesicle will form around it. Now notice that sometimes what we're engulfing is a big particle, sometimes it's tiny particles. If the particle is large, we're going to use a vacuole. If it's small, we're going to use a vesicle. The only difference in these two is size. Vacuoles are larger. But we will engulf, keeping our plasma membrane intact so we don't risk the cell losing anything. We will pinch off a vesicle or a vacuole and bring in either a large product or little tiny products. If we bring in large products, we say we have undergone phagocytosis, because phage means to eat. It means to bring in something large. Penocytosis literally means to drink, so it's bringing in liquids or tiny small particles. There is a third form of bringing material into a cell, and it is called receptor-mediated. Now, it is penocytosis, but it's a specific kind where our little pit that we're going to bring things in has receptors, and instead of just randomly bringing in material like we saw here, instead of just bringing in these little random dots, we're going to specifically only bring in not only materials that fit the receptor, but only the number of materials for the number of receptors we have. Whereas in this first example of pinocytosis, we might bring in hundreds. We might only bring in 10 or 15 here. It is a special form where the vesicle is bringing in specific molecules in specific quantities, like vitamins, hormones. We want to regulate the entrance of those into a cell. It's much more selective. It's also much more efficient. In passive transport, materials move from high to low without the expenditure of energy. Osmosis is a particular type of diffusion in passive transport. Facilitated transport is also passive. And then we have the two forms of I need energy, active transport in general, and then a specific form referred to as bulk transport. Now our cells occasionally will need some modifications. We have a plasma membrane. It is selectively permeable. It does have proteins that can allow materials in and out. It also has cholesterol to stiffen it. It has ID tags, those glycolipid, glycoprotein chains. But we're going to need to add some modifications to that. In an animal, we have what's called the extracellular matrix. The extracellular, literally meaning what is found outside of the cell. What is the material outside of the cell? What is the background? Well, outside of a cell, we can have quite a bit of things. We can have polysaccharides, those large sugars. We can have proteins outside of the cell. Mostly we have collagen, which is a really white, flexible fiber that doesn't stretch very well, but it does allow these cells to work together in a flexible fashion, like your skin. Your skin has flexibility because it has collagen in it. Elastin. Elastin is going to give resilience. Now, think about elastics. A lot of times we unfortunately think elastics as only the ability to stretch. But if you have something that is elastic and it loses its elasticity and you go to stretch it out, it breaks. It loses its recoilability, its resilience to stretch and snap back and stretch and snap back. And that's what elastin will do for our cells. We also have fibronectin, which is going to bind to a protein called integrin and assist the communication between what's outside of the cell, the extracellular matrix, and the skeleton inside of the cell. We also have protoglycans. Prota, protein, glyca, meaning a sugar, that keeps your extracellular matrix from compressing, from being squished together. It gives it some volume, some resistance. It also assists in the cells signaling each other. So we have background, we have material found outside of the cells. We also have the ability to put cells together. In animal cells, we can actually stick one cell to the next cell by one of these three ways. We can have an adhesion junction, sticky. A desmosome is its proper term, or we may have a half one called a hemidesmosome. This is like a snap, like you'd have a snap on your blue jeans, where we're going to literally snap that flap to that flap. But now, unlike a snap on blue jeans, these are not going to come apart with a little bit of tension, because these have these long filaments that anchor very deep in the cell, holding the cell together. This allows two cells to undergo quite a bit of stress and pulling and tension without snapping cell A away from cell B. Your heart, the stomach, the bladder, the skin, these all undergo great stress. Think about the pulling and tugging we do in our skin, the continual filling and emptying of your stomach and your bladder, and the constant stress the heart is under. We need an adhesion junction or a desmosome to keep those cells snapped together so that these cells are making a tissue. We also have a tight junction. Now, a tight junction, if this is kind of like a snap, we want to think about this as like a Ziploc bag. So think about a Ziploc. Now, not a cheap Ziploc, but a regular good Ziploc bag. If you open up the top of a Ziploc and look on one side, you'll notice that there are grooves. If you look on the opposing side, there are ridges. and that ridge fits into that groove and forms a seal. So let's consider why we would use a Ziploc. Most often we put food in a Ziploc. We're wanting to keep bacteria out, keep the air out. We want to keep the material in the Ziploc bag fresh. But that's not the only reason we would use a Ziploc bag. When you travel, you may choose to put your shampoo down in a Ziploc bag. Now, you're not trying to keep anything out of that. You're trying to keep the shampoo in the bag in case it spills. We use Ziploc bags to keep things in, and to keep things out. This acts like a giant Ziploc around cells, forming an impermeable barrier. Your intestines has digested food in it, but it also has a lot of waste in it. We don't want that waste to leak out into the body. Your kidneys, we don't want the material to leak out into your body. So we seal the cells that make up the kidney, that make up the intestines, with a tight junction, a zipper-like junction that won't let material in or out. All right, so an adhesion junction's to hold things together, a tight junction's to seal, a gap junction, which is also sometimes called a communicating junction, is what's going to join two cells. Cell A to cell B can actually let material pass through this gap junction. Now, this is like a little coffee straw. Not a big fat McDonald's straw, but a little tiny coffee straw. The size is important because the size is a restrictive nature. It will allow some things to go through, tiny solutes, glucose, sodium, but yet not allow whole entire organelles to move from cell A over to cell B. Cells that we want there to be communication, like the heart, we want it to work in unison, work together. Smooth muscle, you want to work in unison, work together. They would have a gap junction. Now these are all in animal cells. We have a desmosome, a tight junction, a gap junction. Animal cells have material outside of their cells like fibronectin, elastin, collagen. But when you look at a plant cells don't have as many modifications because their cell wall is permeable, freely permeable. It lets materials in and out. You want to treat a plant, you can go and dust it with a chemical and it will absorb that chemical. If we want to be treated, there are some chemicals we can just put on our skin, but most we have to actually ingest. This freely permeable cell wall does have cellulose inside of it. Cellulose is the stiffening agent, helps give the plant cell wall some structure. There is a point at which there's a penetration between the cell walls. It's referred to as a plasmodesmata. Now the plasmodesmata has little strands of cytoplasm, but they're little openings. That allows water to move from cell A to cell B. It allows glucose to move from cell A to cell B, oxygen molecules to move back and forth. It's kind of like our gap junction in an animal cell. But this goes not only through the plasma membrane, but also through that thicker, tougher cell wall. So if we consider the membrane of a cell, we refer to it as a fluid mosaic. It's made up of many tiny pieces like mosaic tile. We have protein, we have collagen, we have a lot of variety. We looked at the different types of proteins, the junction proteins, the carrier, the channel, the enzymatic, and they each have a unique structure because they each have a unique function. We also talked about the permeability of the plasma membrane, the ability to move things not only from high to low, which would be passive transport, but sometimes in the reverse mechanism, moving things from a low concentration to a high. That is active transport, and that will require the expenditure of energy. Then we also looked at the modifications to these cell surfaces. Particularly animals have more modifications. We have an extracellular matrix and junctions between our cells, and then the plant cell with their plasmodesmata.